Hello everyone and welcome back to Molecular Biology Review. I am your host, Ryder. Today we are not messing around. We're going to look at some real new February 2017 genetics highlighted article investigation. It's some real molecular genetics for you guys and I know you won't be disappointed. The paper is called Evidence for Amino Acid Snorkeling from a High-Resolution In Vivo Analysis of Fis1 Tail Anchor Insertion at the Mitochondrial Outer Membrane. It's done by a group in Istanbul, Turkey, by the names of Keskin, Akdoğan, and Corey Dunn. So basically this is something that really interests me. This is basically a protein chemistry 3D structure investigation conducted completely through genetics, not using any real biochemistry. Basically what they're going to do is they're going to use genetics, knock out parts of genes, even down to the single amino acid, and then you've got a functional readout. You've got the phenotype of that mutant, and whether it's wild type or mutant is going to tell you whether that residue is important for the protein's function. Alright, so Fis1p, recall that in yeast, proteins are called the name of the gene with P at the end to denote that it's the protein. So Fis1 is the gene that produces the Fis1p protein. Never mind what it does, but it's known that the tail anchor of this protein is necessary and sufficient for insertion of the polypeptide into the mitochondrial outer membrane. However, the mechanism is not known. It's been suggested to reach a final topology in the outer membrane in which the amino terminal protein faces the cytosol, which is the outside of the mitochondrion, and a very short and positively charged carboxyl terminus protrudes into the intermembrane space, and the two being connected by a membrane anchoring domain. So basically what they're going to do is develop their screen. Well, selection is a more potent kind of a screen. A screen allows you to determine which things are important, but a selection gives you only those things that are important, and the rest of them die. So in developing their selection for TA mutations that diminish Fis1p targeting, they reasoned that fusion of a transcription factor would lead to insertion within the mitochondrial OM and a lack of nuclear function. Right, okay, so they're going to fuse a transcription factor to this anchor protein. The transcription factor is going to be stuck in the mitochondria and it's not going to be able to translocate to the nucleus. However, then if they mutated those residues in the tail anchor, then Fis1p fused to a transcription factor would escape and it would get mislocalized and it would go to the nucleus. And then they could basically attach a DNA element to that, like it's yeast, so it's probably a growth on some kind of nutrient. So what do you know? They say that there's already S. cerevisiae yeast strains allowing titratable selection based upon nuclear entry and subsequent binding to Gal4P responsive DNA elements, which are readily available. So what they're going to do is use Gal4, very classic transcription factor, and fuse it with Fis1. Oh, and they're going to even put GFP in there. Why not? So what did I tell you? To assess failed Gal4, GFP, Fis1p targeting to mitochondria, we specifically took advantage of the Gal4P-driven His3 and Ura3, that's histidine and uracil, oxotrophic markers in a strain commonly used for yeast 2 hybrid assays. Similar to cells containing an empty vector, Gal4P fused to Fis1p was unable to allow proliferation on medium lacking histidine and containing 20 millimolar 3-AT to competitively inhibit any His3p produced independently, or in medium lacking uracil. However, the same Gal4 GFP Fis1 polypeptide devoid of its TA, called delta TA, permitted ample proliferation on the same two selective media. So that's pretty much their first validation of their model. Here's the result. Here's the yeast results are so beautiful. Here we go. It's right here that we want. So read across. 1, 2, 3. So the first one, basically their control with empty vector, can grow on normal medium, cannot grow without his, cannot grow without Ura. And the second one, GAL4, with the tail anchor in, can grow on normal medium, cannot grow on his, cannot grow without Ura, without his. And third, when they take that fusion protein, but then they delete the tail anchor, well then it can grow in the absence of these two nutrients because the protein escapes the mitochondria, it's fused to a transcription factor, it goes to the nucleus contains these sequences that allow it to grow on without histidine, to produce its own histidine, and to produce its own uracil. Alright, so we immediately sought mutations in the TA that would block targeting of GAL4, SFGFP, Fis1p to the mitochondrial membrane by isolating colonies spontaneously arising on medium lacking uracil. So that's what you call looking for natural mutants. So they look for natural mutants that would be not revertants, but basically they would look for these spontaneous revertants that would revert from not being able to grow on uracil to being able to grow on uracil. So what they did to kind of find these mutants a bit quicker, they used mutagenic PCR and they generated some altered tail anchors inside that transcript for their GAL4 Fis1 fusion, and then I guess transfected those and plated them into the histidine negative medium. And what they found by sequencing is that they had one clone with a valine to an acid mutation, 2 clones with a leucine to a proline, and 1 with double mutation. And basically here's the description of that. Here's at the top, that's their wild type, so that's with their tail anchor intact, cannot grow because it does not translocate to the nucleus. But then they did some mutagenesis through PCR mutagenesis, and they got these different mutants with valine to glutamate, leucine to proline, and some other ones. And here's their, positive control at the bottom. And basically these are yeast colonies, right? If you've never seen this kind of result, they've got yeast colonies, they plate them out at specific dilutions, and basically it's a test of how well the yeast can grow. So if they can't grow in that medium, you probably won't see anything even with the highest dilution, and if they can grow, well then you might see all 4 spots. So to determine whether these mutant tail anchors actually cause the protein to relocalize to the nucleus, they fuse them instead to M-cherry, a fluorescent protein. So buoyed on by those results, basically what they wanted to do was they wanted to get a library of mutants in the Fis1p tail anchor with every single possible amino acid change at every position. So there are 27 amino acids, so 27, the number of amino acids to the power of 27, 20 to the power of 27, I think. Basically all they did was just a very permissive mutagenesis, which would allow them to get every kind of possible mutant, isolated them all and sequenced them, and then just picked the ones that had a single amino acid change. And they got 99% of them. What they found was they did a bit of bioinformatics and tried to determine whether this tail anchor was genetically conserved through evolution, and basically they couldn't really find that much that was exactly conserved. Only one amino acid residue, a glutamine or a glycine, but rather they basically concluded that there's no consensus sequence. It's just you just need a certain characteristic of the TA. So the TA is alpha helical, so obviously inserting proline, which is tightly twisted inside that, is going to disrupt the alpha helix. Proline doesn't work in alpha helices. So yes, in fact they found that basically placement of proline at many different positions would not work. However, they did find one possible mutation, G137 to proline, was still permissive of proper targeting. So they basically suggested that maybe there's just 2 helices that are separated rather than a single monolithic alpha helix. So here they're presenting their data of the clones that are recovered after you select four nuclear translocations. So you select for the destruction of that helix, and basically, here's the ones. This along this axis is fold change. How many were highly enriched? So this is more times enriched, and on this, it's how many copies, the quality of their sequence. So. And what you find is inside here, well, a lot of things changing to proline, a lot of hydrophobic amino acids like V, A, and L turning to things like charged residues, and likewise with positively charged residues like K and R changing into things with negatively charged residues. Okay, so now we get to the snorkeling part. So basically they said, oh, well, we were surprised to find that positively charged amino acids are inside the membrane domain. But really, it's not so surprising, because the phospholipids are actually negatively charged at their head groups, so you need that positive charge to neutralize the negative charge. And what they suggest here basically is that certain positively charged amino acids, like glycine and arginine, they tend to have really long chains on their side chains, so this will allow them to basically stick up from the middle of the membrane and interact with that group on the head group. There's one more kind of interesting thing. They had one mutant where arginine was basically changed to a stop codon. So arginine at a certain point was changed to a stop codon, so all the rest of the membrane anchor was deleted, and basically it was still able to insert into the membrane a little bit. So I'm not sure exactly what's up with that, but I think they basically concluded that even a partial tail with some positively charged residues can still insert in. It doesn't actually require the entire thing. So in conclusion, proteins localized to mitochondria by a carboxyl terminal tail anchor play roles in apoptosis, mitochondrial dynamics, and protein import. To reveal characteristics of TAs that may be important for mitochondrial targeting, we focused our attention upon the tail anchor of the Saccharomyces cerevisiae Fis1 protein. Specifically, we generated a library of Fis1p TA variants fused to the GAL4 transcription factor, then using next-generation sequencing, revealed which Fis1p TA mutations inhibited membrane insertion and allowed GAL4p activity in the nucleus. Prompted by our global analysis, we subsequently analyzed the ability of individual Fis1p TA mutants to localize to mitochondria. Our findings suggest that the membrane-associated domain may be bipartite in nature, and we encountered evidence that the positively charged patch at the carboxyl terminus is required for both membrane insertion and organelle specificity. Furthermore, lengthening or shortening of the Fis1p TA by up to 3 amino acids did not inhibit mitochondrial targeting, arguing against a model in which TA length directs insertion of TAs. Most importantly, positively charged residues were more acceptable at several positions within the membrane-associated domain of the Fis1p TA than were negatively charged residues. These findings, emerging from the first high-resolution analysis of an organelle targeting sequence by deep mutational scanning, provide strong in vivo evidence that lysine and arginine can snorkel or become stably incorporated within a lipid bilayer by placing terminal charges of their side chains at the membrane interface. Thank you very much for watching. As always, I'll put the links to these articles in the description, and keep studying hard.